Hello everyone, we are ISTANBUL. Today, we are going to present our project, the living laser, transforming cells into lasers. There are some other projects that get lasing out of cells. However, they provide non-biodegradable material to the cell to get lasing. We aim to create a perfectly biodegradable biolaser that only needs the excitation source to get lasing out of it. Creating a fully biodegradable biolaser is crucial since conventional photonic devices are aimed to be replaced by totally biodegradable biolasers at first place. We believe that cells can turn into lasers. They just need a little help from their friends. Creating a biolaser is cool. With that said, it turns out that it's useful too. So, why do we want to make living lasers? When a cell changes in shape, we get a different lasing out of it. Thus, our biolaser mechanism enables us to detect shape changes in different cell types. In future studies, this property of our technique could be used to early detection of cancer cells, since the shape of cells change when they turn into cancer cells. There are more than 2 million people diagnosed with breast cancer each year, and early diagnosis is crucial for a successful treatment. Also, our technique could be used in oocytes, since their shape is quite crucial for in vitro fertilization. Minimum wage is 280 USD in Turkey, and one IVF cycle is 900 USD. So, minimizing the trial numbers is crucial for couples. Another possible application would be early detection of cirrhosis. In our country, health insurance doesn't pay for the treatment unless the patient is diagnosed. So, early diagnosis is vital for cirrhosis patients. How would we achieve that? Without getting into our technique, we can talk about what is a laser and what they've done before to understand the difference with our project. A laser has three fundamental components: a gain medium, a resonator, and an excitation source. A gain medium is the source of optical gain in the laser system. A resonator is a reflective part that helps the system to reach the needed light energy to pass the lasing threshold. Excitation source provides the required excitation energy, which is light energy in our system. This is an ideal system which has a nearly 100% reflective mirror and an 80% reflective mirror. These mirrors trap light inside of the system, which allows light to amplify more and more. In this system, they use fluorescent proteins as gain mediums. However, their resonators are non-biodegradable optical lenses. Also, their resonators are outside of the cell, which means that their cells did not turn into lasers. We are using two cells, E. coli and yeast. There are several reasons for that. They are both cheap and easy to work with. Their size and shapes are ideal to get lasing out of them. Our main goal is to cover their plasma membrane, or for the yeast cell wall, with biosilica and reflectin protein. By doing that, we get a fully biodegradable biolaser mechanism. When we get lasing out of them, we will be able to track the cells and their growth. Single cell tracking is challenging to achieve since using different fluorescent proteins with different excitation wavelengths is not enough to detect each cell. Even if fluorescent proteins have major excitation peaks, their beam is not focused when they are excited with conventional microscopy techniques. That causes signals from single cells to mix and makes it impossible to use more than 4 to 6 fluorescent proteins. In our technique, when cells turn into lasers, their laser radiates in a single wavelength, which allows us to track them cell by cell in real time. By this technique, more than 100 fluorescent proteins could be used without their signals mixing.
There are two types of gain mediums we focalized. The first one is the fluorescent proteins. Fluorescent proteins could be synthesized dyes. We plan to use DIPI, androstenedione green, and acridine orange, since they are optimal to be a gain medium for our technique.
First studies made on biolasers. Eukaryotic cells are used for expressing fluorescent proteins as gain mediums and placing them between a resonator made of two parallel mirrors. After these experiments, it was proven that cells were capable of producing lasing, and later mirrors were eventually replaced by artificial microbeads. Although These microbits had to be replaced by biomaterials in order to create a true biolaser. In regular lasers, mirrors are used to generate a resonator. Instead, we will use natural materials such as proteins and biopolymers that are generated by enzymes. We will use two different proteins to achieve our goal. We will first express silicatein alpha protein, which is found on axial flamellae of the marine sponge, and catalyzes the formation of biosilica layer with a high refractive index that could act as a resonator. Additionally, we will express reflectin A1 protein, which is a protein with high refractive index that is mostly found in cephalopods and demonstrates camouflage capabilities by adaptive transparency. To generate a resonator covering the bacteria, silicatein alpha must be first expressed in cytoplasm, then located to the bacterial cell wall and treated with silicic acid for biosilica layer polymerization. Silicatein is a protein that can go out of the cell and cover the cell wall with the biosilica layer by using a specific protocol involving silicic acid treatment. Although, we will also try binding a membrane-bound tag called OMPA to silicatein and locate more silicatein proteins to the cell wall of the bacteria. Cells will also contain fluorescent proteins that will act as a gain medium. With the complete resonator structure, it will resemble a biolaser ready for lasing experimentations. After experimenting with silicatein alpha proteins, we will express reflectin proteins and locate them into the periplasm of the bacteria. Reflectin proteins are found in cephalopods such as octopuses, squids, and cuttlefish that allow rapid color changes and adaptive transparency for the host. As a result of its high refractive index, it is capable of engaging in structural coloration iridescence. Because of its qualities as such, it's a good candidate for a biolaser resonator. We will use TOR-A and lip-A tags attached to the reflectin proteins to translocate reflectins into the periplasm of the bacteria. With its high refractive index, reflectin proteins will provide efficient layering to trap light beams and generate a precise laser beam in the bacterial cells. Yeast cells are significantly bigger than their bacterial counterparts and therefore can be a good host for generating a functional biolaser. Silicatein alpha and reflectin A1 proteins will be translocated to cell wall by PIR3 tag attached via glycine-serine linker, similar to experiments done with bacterial cells. Additionally, an anchor domain will be used to translocate the reflectin proteins onto the cell wall. Fluorescent proteins will be expressed as gain medium, and the yeast cells will be tested for their lasing abilities.
In our project, we will be using a generic fluorescent microscopy setup for lasing experiments. We will be exciting the fluorescent proteins in the sample using a pulse laser, which can be tuned to any frequency in the visible region. Emission from the cells will be collected by optical elements and directed towards the cyphtograph. We will be imaging the sample using a CCD camera, whereas the cyphtograph will be used to analyze the emission spectrum of the sample. We have used the data obtained from gel densitometry and flow cytometry while creating our biological model. We use the factors of protein expression, quantum yield, molar extinction coefficient, and in vitro brightness to assess radiation from cells. Our innovative part involves the integration of protein maturation into the kinetic model. The general expression of the model is as follows. Maturation factor can be calculated as 1/1 plus T50 over T growth. T50 is the time when the 50% of fluorophores mature, and T growth is the doubling time of the bacteria. We have created computational models of our biological resonators and performed finite difference time domain simulations using numerical FDTD software. By using the time domain method, we can easily simulate what happens inside the cell in terms of optical means. For our project, we have created cell models of 3 different geometries: spherical, toroidal, and disc-shaped. Membrane of the cell and reflective molecules inside the cell are successfully incorporated into our models. Using our computational models, we have revealed the effects of cavity size and the refractive index of the membrane on the whispering gallery modes occurring on the surface of the resonator. Since one of our focus is using these cavities to track cells individually, unique resonance modes corresponding to minute differences in size and optical properties of the membrane is very important to model in our project. We have determined the resonance modes for each cell model type individually. Two main parameters we have checked in our simulations are the size of the cavity and the refractive index of the membrane. For the size parameter, we have run simulations on three different geometries with diameters varying from 1 micrometer to 10 micrometer. For the refractive index, we have run simulations on all of our models by using different refractive index materials varying from 1.6 to 2.5. For our models with 10 micrometer diameter, we have seen very sharp peaks on power spectra graphs. It points out that we can easily observe whispering gathering modes from these models. Although the graphs on the screen belong to membrane indices of 2.0, we have successfully observed resonances for refractive indices of around 1.7. For our models with 5 micrometer diameter, we have nearly lost those sharp peaks on power spectra graphs, especially for refractive indices lower than 2.0. This means that if we will use cells with 5 micrometer or lower diameter, we need to cover these cells with proteins with refractive indices higher than 2.0. Overall, results of our simulations is that we can obtain whispering gathering modes from cavities with diameters 5 micron or bigger using special proteins as a coating material for the membrane of the cell. From this result and the recommendations of the previous IGEN team, Delft 2016, we have included Baker's yeast into our experimental plan since they are much larger than most bacteria and have nearly spherical shapes before they start budding. Also, these results set some boundaries for our initial plan, and we have included much bigger cells into our future experimental plan. We are confident about achieving lasing from resonators built from yeast cells by covering them reflectins and silicatein. Our computational results are satisfactory for our purposes, even they are still lacking some details of real biological cells and their environment.
We use E. coli and yeast as a model organism as they're really cheap, so that we're trying to create a cheap model for future lasing implementation. For instance, we want to detect breast cancer in its early stages. To do that, we're going to insert fluorescent dyes into the breast tissue, and the dye will enter the cells and make them fluorescent, and the cells stimulated by the light will laze due to the fluorescent dye in the resonator. And those lasing will get detected by a detector. But how are we going to differentiate a healthy cell and a cancerous cell? As you can see, a cancerous tissue and a healthy tissue have different properties, and these affect their cell shapes. As we said, these cell shapes affect the lasing that we get out of these cells, so that we can differentiate those signals. We decided to work on breast cancer after we talked with MD Atakan Demir and after we did some background research after the talk, because we first thought that we can work with every cancer type, but after we talked with Atakan Demir, he said that we can't do that because there are many different tumor types. So focusing on a common cancer, such as breast cancer, is really crucial. And also, he said that he can help for ethical committee for working with tissues and cells in the future. And he also suggested that we should work with people who does genetic cancer proneness tests so that we can implement biosensors into those people in the future. After we talked with MD Atakan Demir, we decided to find a company that does these cancer proneness tests. And we found Genetics. What they do is basically they do BRCA1 and BRCA2 tests for breast cancer, and they do 250 gene tests for other cancer types. And after we talked with them, they said that we can work in their R&D department in the future about this project. And they also said that we can't do tests for under 18, so we should work with people who are over 18 in the future. Also, in the future, we want to determine the quality oocytes by using their lasing properties, as they have natural resonator properties outside their layer. So the shapes of the oocytes determine if they can laze or not, so we wanted to harness from these skills of these oocytes. To discuss about our idea, we talked with Dr. Simopoulou from Greece, thanks to Athens IGEN, and we talked with Dr. Kayhan Yakın from American Hospital in Turkey. Dr. Simopoulou told us about how to use lasers in IVF centers and what we can do with our project and how we can focus our projects in detail. So she generally said that we should focus on the stimulation protocols of oocytes in determining quality oocytes. And she also told us that we should focus on creating and characterizing these oocytes and creating a correlation between how they affect the IVF cycle and if we can blame the quality of oocytes if there is an unsuccessful IVF cycle. And we also talked with Dr. Kayhan Yakın. He also told us about how these kind of studies work in Turkey and how we can study with oocytes. And he also told us about how the studies with embryos are also important. And another implementation that we thought can be useful is the degradation of ICG dye by liver, as we can look at this ICG and we can track it as it can laze inside the blood, as blood also have a natural resonator property. If the liver has some impairment like cirrhosis, it will degrade this ICG slowly. So after degradation, we won't get any lasing so that we can calculate how much time does it take to degrade this ICG in the liver. And we discussed with MD Esaryanik about this idea, as he also suggested this idea to us after we talked with him. And he also said that we can use an FDA-approved drug such as ICG. And we also discussed about the safety aspects, and he said that if we can create a proof of concept with yeast and E. coli about lasing, then he thinks that these biolasing structures can be used in many other ways, such as this liver project. And he also informed us about creating a startup in Turkey. So we're also included this in our plans in the future so that we can create a startup by using the big grants in Turkey. And another implementation that we thought about is actually determining the glucose concentration in blood by using aspirin orange and the natural resonator properties of red blood cells. As aspirin orange binds directly to hemoglobin, and hemoglobin binds to the glucose, we know that we can get lasing out of these red blood cells because of their properties too. When the glucose concentration changes in the blood, the shape of these blood cells change too, so that we can detect different lasing from different shapes and thus different glucose concentrations. And another application that we thought is creating a live biosensor for airborne transmitted viruses such as SARS-CoV-2. We can do that by using the natural resonator properties of oocytes. And we are planning to engineer some mutant oocytes that can detect those SARS-CoV-2 viruses. They can bind directly to our oocytes, and when they bind to our oocytes, they will get activated by GFP, and that GFP will get laze because of the resonator properties of the oocyte. And when the GFP lazes, there will be a detector at the back of the head that can detect the lasing so that we can understand if there's a virus or not. And this project is funded by TÜBİTAK, so we know that it is safe in the future. The last application that we thought is creating a system for Human Cell Atlas project. Human Cell Atlas project focuses on characterizing every single cell in humans, but they have some difficulties as it is hard to differentiate the signals when you use fluorescence microscopy and fluorescence in general, because the fluorescence spectra is large and there are some overlappings. But if we can make these cells laze, we can detect just 1 nanometer of signal from every cell so that we can differentiate those signals and thus we can track them and we can do single cell tracking. So this is one of the biggest applications that we thought, as if we can check every single cell by using our technique, this means that we can track millions of cells at the same time.
After we complete the proof of concept on yeast and bacteria, we will repeat lasing tests and proof of concept tests on red blood cells and also on the atoms, since they have natural resonator properties. We will basically put fluorescent dyes inside the cells and try to get lasing from them. Also, just like we said, we are planning to use oocytes as well. We are planning to use them with different dyes and without dyes. Our plan is testing them with fluorescent dyes, fluorescent proteins, and without both of them with their natural resonator properties. So at the end, our team wants to thank to these companies and honorable scientists for all of their amazing support. Thank you on behalf of the team.